always, a few announcements. One is today's the first day of spring. Another is that quiz 3 is due on Sunday, and it focuses on anatomy of sensory systems. It will not get into the leg muscles that we're starting to work on in lab. That's not on the quiz. And it's not doing motor anatomy. It's doing sensory anatomy. Somatosensory, vision, auditory, taste, smell, fraction. Anatomy of those systems. The key concepts list has the names of the things that you need to know for the anatomy of those systems. I'm happy to answer questions between now and Sunday. We are doing a review on Tuesday for exam 2. So exam 2 is a week from today. Tuesday will be a review session. I'll have a bunch of, I don't know, fill in the blank sort of things, or maybe we'll play the anatomy game, or maybe we'll do some, probably a bunch of clickers, but that's just to get the ball rolling, the ball being you asking questions and me answering. Okay? So these are back to back, right? I mean, quiz and then the exam follow pretty close upon each other. And so the idea is that the quiz and the exam, this is part of a unit, right, test. The quiz will focus on anatomy. The exam will focus on physiology. But the exam might throw in an anatomy question because anatomy informs physiology and the other way around as well. So the exam will mostly be physiology, but with a little bit of anatomy thrown in. Certainly the legs and the motor anatomy, which wasn't quizzed on the quiz, could be examined on the exam. Question? If you're in Econal and you have an exam on Thursday, March 28th, that's a good reason to get an extension on the A&P exam. If you have another thing that is due on that day, that's all, similarly, I would give you an extension. So if you are in Econal and you have an exam on that day, or you have some other deadline that day, I'm not talking about that week, I'm talking about that day, then let's talk after class or at the break and come to an agreement on an alternative day for your exam. Because taking the Econal exam and the A&P exam on the same day is a cruel and unusual punishment. And the final announcement is, this is sort of a last call for Brain Explorers. If you're interested in participating in Brain Explorers as a presenter, talk to Mallory after class today. And if you do participate, you get this t-shirt. That's part of the drill of participating in Brain Explorers. So a neural network, so in the center there is the heart. Right? Question? Yes.
For the exam, are we just doing it in class, or are you going to offer it again at the lab?
It's in class. I can't, so that's not a lab day, so I haven't decided whether to offer it in the afternoon. I have another class at 2:30, so I can't be here for the full afternoon, but I suppose I could do it at 1 o'clock. I'm not teaching at 1 o'clock on Thursday. So if you want to explore an option of taking it at 1 p.m. on Thursday as opposed to 8:30 p.m. 8:30 a.m. on Thursday, let's talk after class. I guess that seems reasonable to offer that as an option, even if you don't have the conflict. I'm open to it. 8:30 is pretty early. All right, so other questions about the sort of rapid fire quiz exam scenario.
In the coloring book, you have like the 5 muscles. Those won't be on the quiz.
Thigh muscles will not be on the quiz, even though they've been assigned
Yeah.
before the quiz, they are not on the quiz.
Okay.
Thigh muscles could be on the exam. All right, so this is the same slide that we got started with yesterday, Tuesday. Tuesday we focused on motor neurons and skeletal muscles, and now we're going to focus on spinal cord and brain, the motor system. So almost none of this will be reviewed from cell physiology. It's all going to be pretty. So you'll remember that a motor unit is the group of muscle fibers that is innervated by a motor neuron. So single motor unit is the set of muscle fibers of a muscle innervated by a single motor neuron. Each motor neuron innervates many muscle fibers, several too many. Each muscle fiber is innervated at one place, one neuromuscular junction, by one motor neuron. So 2 motor units, 1 muscle, but 2 sets of muscle fibers in that muscle, 2 motor units, 2 motor neurons innervating selected muscle fibers, 2 motor units. The motor neurons, the neurons that innervate skeletal muscle, have their cell bodies and their dendrites. They receive their input information in the ventral horn of the spinal cord. So let's get oriented here. This is a cartoon of the spinal cord in horizontal section. So this is anterior, this is posterior, this is left, and this is right. Okay, good. Remember to get oriented. So here is the dorsal horn, sometimes called the posterior horn of the spinal cord. That's where sensory information comes in and where the first synapse of the pain or temperature pathway is. Here is the ventral or anterior horn. I go with dorsal horn and ventral horn, but posterior and anterior are accepted. So this, the ventral horn is where the motor neurons, cell bodies are, and then they send their axons out this ventral root that then merges with the input axons coming in the dorsal root. There's the dorsal root ganglion. And so one nerve, let's say your median nerve in your arm, by the time we get out here, is carrying both motor information from the spinal cord and sensory information to the spinal cord. But the sensory information comes in on the dorsal side, and the motor information goes out on the ventral side. So here's the cartoon. There's some information here about that you don't need to worry about. This is actual data. This is a stained section of a spinal cord. So here's the dorsal horn, here's the ventral horn, here's the dorsal root ganglion. So here are the axons coming into the dorsal horn, and here are the axons going out the ventral horn and forming then a merged nerve, sciatic nerve, femoral nerve, median, some nerve that is innervating skin or muscle. Receiving information from skin, providing motor information to muscles. So again, the ventral horn is where the cell bodies are for motor neurons. There is an organization of the ventral horn. The more medial cell bodies innervate proximal muscles. More lateral cell bodies innervate distal muscles. So the more medial you are in the ventral horn, you being a motor neuron, the more medial the motor neuron cell body is in the ventral horn, means it's going to innervate proximal muscles. And so if this must be a section through the cervical spinal cord, because these motor neurons are innervating the arm. In the lumbar region, the medial motor neurons would be innervating the proximal muscles of the leg. The lateral motor neurons would be innervating the distal muscles of the leg, toes, toes and ankle and stuff. So there's an organization of the motor neurons in the ventral horn, and there's actually a motor organization where the extensors are more ventral and the flexors are more, central in this organization. So we have a sort of a two-dimensional array, right? Medial, lateral, proximal, distal, and ventral, dorsal, extensor, flexor. I will not ask you to memorize this map. I have asked you to memorize certain other maps, like where the high-frequency sound is mapped on the cochlea and where the high-frequency sound is mapped in the auditory cortex. This one, I won't ask you to memorize, but I might ask you to solve a problem with this if I give this information to you. And you can see, and then moving on, you can see that the amount of gray matter varies as a function of the different levels of the spinal cord. So in the thoracic region, because there aren't a lot of thoracic muscles, there's relatively small ventral horn, relatively small gray matter. Whereas in the lumbar and the cervical, where the motor neurons are innervating leg and arm muscles, of which we have a lot, we have a relatively large gray matter. And in the sacral as well, because that's also innervating as part of the leg. Okay, so now we've talked about where the motor neurons are. Now what we want to do is talk about something called central pattern generators, which are important for the motor system. So it turns out that a lot of our motor coordination, now we're not just talking about one motor unit contracting one muscle. We're talking about motor coordination that gives us the ability to walk and execute specific programs of movement. And a lot of that information is not in the brain, it's in the spinal cord. The central pattern generators that coordinate just sort of stereotyped movement, like walking, or swimming if you're a swimmer, or, I don't know, throwing a frisbee if you're a frisbee player. Certain programs that you run during your life as a human being. A lot of that information gets stored in a program in your spinal cord, not in your brain. Particularly walking, that's the one we've evolved to do. But I suppose throwing, right? Because when we were predators, throwing a rock or a spear or a dart or something to catch a prey, that would be something that we've evolved to do. Running as opposed to walking, that's something we've evolved to do. Those are motor programs, and those are resident in the spinal cord as central pattern generators. So let's talk about that for a little bit. So the simplest way, so what I want to do is build this concept of a central pattern generator that is in the spinal cord. And the first way to build this of these programs that are in the spinal cord that control muscle movement is to talk about a couple of reflexes. So the first reflex is one that you might be aware of, called the knee-jerk reflex. You sit on the doctor's bench, the doctor or the physician's assistant or the nurse takes a little hammer and hammers on your patellar tendon, and your knee jerks in response. That's the knee-jerk reflex. So let's unpack that a little bit with the knowledge that you have from anatomy and physiology. So let's get oriented. Here's the spinal cord, here's the dorsal root ganglion, here's the dorsal root, here's the ventral root. So sensory information is coming in the dorsal root, and motor information is going out in the ventral root, and it's innervating flexors and extensor muscles. And remember, remember, that muscle, we have antagonistic muscle control. This is an important. We have flexors and we have extensors, and they work against each other, right? When I contract my flexor, I'm relaxing my extensor. It would be pointless for me to contract both my flexor and my extensor at the same time. That would be a waste, because I wouldn't move. That would be, the extensor and the flexor, the extensor is trying to do this, the flexor is trying to do this. In order for me to do this, I can't be contracting the extensor at the same time. They work against each other to extend or to flex. And so in order to have efficient flexion, I need to contract the flexor and relax the extensor at the same time. In order to extend, I need to contract the extensor and relax the flexor at the same time. That's part of this central pattern generation. So let's walk through this one, the knee-jerk reflex. So here's where, this is from your book, and it walks through pretty clearly. Let's look at the beginning, where the physician or the nurse or the PA taps on the patellar tendon. The patellar tendon is connected to the extensors, the quadriceps, which are the extensors of the knee, and stretches it. So muscle spindles, that's what this spiral is supposed to represent. Muscle spindles in the extensor muscles get stretched. And they fire action potentials to the spinal cord, that go to the spinal cord, informing the spinal cord, oh, my extensor muscles here in the quadriceps are getting stretched. That's information that the body needs to know. So now let's look at this synaptic connection. Remembering that a green axon is an excitatory axon, and a red axon, a red neuron, is an inhibitory interneuron. You've heard those words before, inhibitory interneurons. For example, the horizontal cells and the amacrine cells of the retina are inhibitory interneurons that do lateral inhibition. So we've talked about inhibitory interneurons before. This isn't lateral inhibition, but it's inhibition. It's an inhibitory interneuron. Doesn't have a very long axon, but it's got an axon. So here, the patellar tendon gets stretched. Stretch proprioception information is coming, that's proprioception, right? Stretch of a muscle is coming in the ventral horn, and the brain is being informed. The brain over there is being informed by the axon, by a branch of this pathway going up the ipsilateral dorsal column to the brain. That's information going to the brain, but that's not what we're focusing on. What we're focusing on now is the fact that this neuron, in addition to informing the brain, makes excitatory synapses with motor neurons of the extensor, and makes it excites the inhibitory interneuron. So that's an excitatory. B is exciting the inhibitory interneuron, which inhibits the motor neurons of the flexors. A simple circuit. At the same time, this input is exciting extensors and inhibiting flexors. So you tap on the knee, and even without thinking, even before your brain has a chance to even respond, the muscles, the extensors contract and the flexors relax, and your knee jerks. That's a motor program, a very simple motor program in the spinal cord, and it's the knee-jerk. It's called the knee-jerk reflex. It's a reflex because your brain doesn't need to be. It does not participate. Israel.
The same scenario like we were just doing at high, so.
Okay, that's the next slide. So it's a similar scenario, but it's the next slide. So questions about this one's simpler. The hot stove is a little more complicated. Questions about the simple motor program. this simple central pattern generator. Remembering reciprocal control of flexors and extensors. When we contract the extensor, we relax the flexor because of this inhibitory interneuron. Okay, let's answer Israel's question. Here is the pain reflex. So he was referring to the pain of touching a hot stove, but in this slide it's the pain of stepping on a tap. Same idea. But we're now we're in the leg. We're staying in the leg as opposed to the arm, but the same concept. So let's walk through this central pattern generator. Pain, afferent nerve, going afferent nerve at cell body in the dorsal root ganglion, information going into the dorsal horn, first synapse crossing over. Okay? You've learned that. Let's not worry about going to the brain. Well, there's the information going to the brain in the contralateral anterolateral pathway, anterolateral tract. But we're not talking about the brain. We're talking about the central pattern generator in the spinal cord. Okay, so here's the information coming into the ventral horn. Synapses, so it makes an excitatory synapse with maybe some excitatory interneurons or maybe not. Let's look right here. It makes an excitatory contact, green, onto the motor neurons that innervate the ipsilateral flexor. And it uses an inhibitory interneuron to inhibit the ipsilateral extensor. So, going to Israel's question, you touch a hot stove, and before your brain even senses pain, your flexors contract and your extensors relax, and you withdraw from the pain. That is a reflex, a pain withdrawal reflex. You've all experienced it. You touch something so hot that your hand is away from it even before you have a chance to feel it. And you might have been burned, but you don't feel the burn. You're not aware of the burn until well after your muscles have caused this reflex to get your hand away from the burning stimulus. Same thing with a tack. You step on a tack and your leg withdraws. You're contracting the flexors and relaxing the extensors to cause withdrawal. Okay? So this is like the so far it's like the stretch, the knee jerk reflex. An inhibitory interneuron, one inhibitory interneuron. It's reversed because we are contracting the flexors and relaxing the extensor on the ipsilateral side. But now let's make it more complicated, because it is. In addition to what's happening on the ipsilateral side, we've got a neuron that crosses over and makes an excitatory connection with the contralateral extensors and makes, via an inhibitory interneuron, an inhibitory connection, a relaxation connection, to the contralateral flexors. So this is start of a motor program. In the hand, it doesn't make a lot of sense because we're standing on our legs. But on the legs, it does make sense. If you contract your leg, you don't want to fall over. We have a program in the spinal cord, a reflex, that causes the other leg to extend. So we're going to flex one leg and we're going to extend the other. In order to extend the other leg, we need to contract the extensors and relax the flexors. So that's what we're doing. So when I step on a tack, I reflexively, without thinking about it, withdraw the ipsilateral leg and extend, that is to say, flex the ipsilateral limb and extend the contralateral. So if you may not even be aware of it, but if you hit a hot stove, you withdraw, you flex the ipsilateral limb, and you extend the contralateral limb. I mean, we can probably override that one, because you're not aware of doing this, but it's still happening. reciprocal control. So is this program, this is central pattern generator that is resident, this pattern of excitation and inhibition is resident in the spinal cord, in the ventral horn. The brain's not part of it. The brain's getting the information about the pain after. It takes a few milliseconds for the brain to even process that information. Is this clear? The idea of the spinal cord resident circuits that generate central patterns. Locomotion is a central pattern generation. So here is a cartoon of a lamprey that has a beautiful swimming motion. Beautiful S-shaped swimming motion. And yet that swimming motion is a result of coordinated excitation and relaxation, or excitation and contraction and relaxation, excitation and inhibition in the spinal cord along the muscles of the body. So for example, motor neuron 1 and motor neuron 3 are alternating. When motor neuron 1 is firing, motor neuron 3 is not. And when motor neuron 3 is firing, motor neuron 1 is not. Alternating contraction. When motor neuron 1 fires, and then a few milliseconds later, motor neuron 2 fires. And then a few milliseconds later, motor neuron 3 fires, and then a few milliseconds, motor neuron 4 fires. So that gives this animal the possibility of making this beautiful S-shaped swimming motion. That's how it moves. And that movement is that pattern of excitation. This patterning of excitation is resident in the spinal cord. We have something similar, but it's more complicated. So we have a rhythm generator, just like in the lamprey, generating 1, 2, 3, 4. We can generate a rhythm of contracting flexors and extensors in order to walk. We have left-right coordination that's going back and forth. So what we're doing with our left side, we're not doing with our right side at the same time. We're going to do it later, in order to move one leg and then the other leg. The motor neuron is the output, but this is the central pattern generator for locomotion. It's in the spinal cord. And then, there's what is intersegmental coordination. When I'm walking, I run a program that controls my legs. First my right, then my left, then my right, then my left. But I also move my arms, right? You're aware that when you walk, your arms swing as well. And it's not just the force of gravity, it's because we're quadrupeds, evolutionarily, and we have alternating contraction of the arms even when we're just walking. And so that's going from the lumbar region up to the cervical region to control the movement of the arms while the legs are working. So trying to understand at the neuronal circuit, at the individual neurons, is too complicated for this. We can do that with something as simple as the pain reflex, which is the starting, right? So remember the pain reflex of this reciprocal control on one side and then reciprocal control on the other side. That's like locomotion. Flexing one leg and extending the other leg, and then flexing the other leg and extending the first leg, right? So that's starting to sound like locomotion, and that's what we've got. So to show this to you, I'm going to show you a slightly upsetting video, which is hard to see because it's really old. People don't do this very much anymore because it's already been done. There's no point in doing it again. So this is a cat that is decerebrate. All the connections between the pons and the rest of the brain have been severed. So this cat no longer, it can still breathe because the breathing centers are in the medulla. it can't sense its body, and it can't control, it has no cortical control of movement. Decerebrate. And you can say, well, that was cruel. But this experiment allowed us to determine that we have these motor programs resident in areas of our body below the brain. That's what it allows. It's separating the brain from the body. And look what this cat can do now. So this cat is being suspended because some of the postural muscles, it's not normal. Obviously, it doesn't have a brain. But so it's being suspended, and it's on a treadmill. And the treadmill is controlling the speed at which it's moving its legs. But watch what you can imagine. Okay, if this treadmill is going to move, and the legs aren't going to do anything, it's just going to drag its legs, and it's going to, just be dragged. The treadmill is just going to drag the limbs, but that's not what you're going to see. What you're going to see is coordinated movement. And in fact, the faster the treadmill goes, the different steps the cat walks, and then trots, and then gallops, all without a brain. Okay? So that experiment, which was bad for that cat, but was really good for us, was to determine the fact that this motor program of walking and trotting, these motor programs of walking, trotting, and galloping existed below the level of the brain. And in fact, they exist in the spinal cord. We could get into a debate about the value of animal experimentation. There are ethical issues. That cat was certainly anesthetized during the procedure, but was not anesthetized, obviously, during the test. That cat is feeling no pain because it has no feeling, period. It's got no brain. So, we've talked about muscles on Tuesday. We've talked about motor neurons on Tuesday. We've talked about central pattern generators in the spinal cord just now. And now what we're doing is moving upward, up into the brain, the cortex, the basal nuclei, and the cerebellum, that control, both directly and indirectly, motor neurons by controlling the central pattern generator. So I can say, walk. My brain is saying, okay, Bob, time to walk. But now my spinal cord is executing the program of walking. My brain is not saying, okay, left, now right, now left, now right. My brain's not doing that. My spinal cord is doing left, right, flexor, extensor, move your arm. Yeah, when I think about it, I can't do it. But if you don't think about it, your arm swings opposite your legs. So now let's go into the brain. Superior view, front, back, right, left. Longitudinal fissure, central sulcus, somatosensory cortex, motor cortex. The motor cortex is the most posterior gyrus. It's in the pre-central gyrus. It's the most posterior gyrus of the frontal lobe, whereas the somatosensory cortex is in the post-central gyrus, the most anterior gyrus of the parietal lobe. But they're right next to each other. So the motor cortex, which participates in the control of the body, but doesn't, and runs the motor programs, but isn't where the motor programs are resident. It's not where the motor programs are, but it's, you know, it's where that cat didn't decide to walk or trot or gallop. That was determined by the treadmill. We decide whether to walk or not, whether to lift our leg or not, and that is part of our cortical control. And the motor cortex, so this is a coronal section, medial, lateral. This is the right hemisphere, so you're looking at somebody's right hemisphere. So this is the longitudinal fissure here. Here is the lateral fissure there. This is the temporal lobe. What lobe is this, if we're talking about the motor cortex? Frontal lobe, the most posterior gyrus of the frontal lobe. And you can see it's got an organization that mirrors the organization of the somatosensory cortex, with legs and trunk muscles mapped medially, and fingers and arms and face muscles mapped laterally on the motor cortex. That is a map that, because it's true for the sensory and for the, I'm not gonna ask you to know, you know, if I say point to where the part of the cortex that controls the lips is, I'm not gonna care whether you point here or here or here, as long as you don't point here or here or here. So we have a motor cortex that controls the motor programs and controls specific movements. Coronal section, left, right, superior, inferior, medulla, midbrain, well, this would be the pons, I suppose. Is the bulge? I don't know, it's a cartoon. I didn't draw it. I'm trying to use some of the anatomical phrases that you have learned for exam 1, and hopefully have not just jettisoned completely for exam 2. Longitudinal fissure, lateral fissure. What is the red? The red is a cartoon of the axons that go from the motor cortex, go through that structure that we named at one point called the internal capsule between the thalamus and the putamen. The internal capsule, and then form what is called the corticospinal tract. Corticospinal tract is the most important set of axons from the motor cortex to the spinal cord, controlling movement, controlling those motor programs, and controlling specific movements. My cortical decision to flex my arm. So I'm not running a motor, I suppose I'm running a motor program in the sense that if my brain says flex your arm, it's saying contract your flexor and relax your extensor. So there's part of a motor program being run there, but my brain's not, I don't have to think about, okay, in order to flex my arm, I've got to contract this muscle and I've got to relax this muscle. Some of it is unconscious. I know that in order to, my spinal cord knows. My brain says, flex your arm. My spinal cord says, okay, that means contract the flexor and relax the extensor. And that information from the brain to the spinal cord is the corticospinal tract. The corticospinal tract is, this is an amazing, relatively new imaging technology called diffusion tensor analysis. So this is actual data. This is not a cartoon or an artistic rendering. So there's a way of being able to map specific axons using color, but it's not, the computer is actually tracking individual axons and then giving them a color. So you can see the different colors of the corticospinal tract going from the motor cortex through the internal capsule, joining together in the pons. In the medulla, there is the decussation, the crossing over of the corticospinal tract. So many, now it turns out not all, but many of the neurons of the corticospinal tract cross over. So most of the control of the left arm starts with, at least at the cortical level, starts with cortical processing in the right motor cortex. Left motor cortex, let's do right motor cortex, crossing over to control the movements on the left side of the spinal cord. Left motor programs, left muscles. Just like the pain pathway crosses over in the spinal cord, and the touch pressure pathway crosses over in the brainstem. The motor program, most of the motor information crosses over in the brainstem. And that's why the right motor cortex is most important for controlling the left side of the body. There are several parts of the corticospinal tract. And I only want you to know about the lateral corticospinal tract. Let's not worry about the anterior corticospinal, and let's not worry about the non-corticospinal. It's actually extrapyramidal, called the rubrospinal and the reticulospinal and the vestibulospinal. There's, motor control is complicated. Let's not worry. So, what does the word extrapyramidal mean? Okay, extra, outside, like extramural or extroverted. Extra, outside the pyramidal. What pyramid are we talking about here? The corticospinal tract is also called the pyramidal pathway or the pyramidal tract. So like so many things in anatomy, it's got two names. Lateral corticospinal tract is also known as the pyramidal tract. There's a part of the medulla called the pyramid. That is where the corticospinal tract is. And so there's some of it outside the pyramidal tract. Okay? We're not going to talk about it anymore. We're going to talk about the lateral corticospinal tract. And the lateral corticospinal tract is in this red area of the spinal cord. So you've seen this image before when we were talking about sensory systems. Blue is sensory, red is ascending, things going to the brain, sensory information going to the brain, and blue is motor, descending, information coming, no, red is descending. Red is information coming from the brain going to the spinal cord for motor control. And right here is the lateral corticospinal tract. And again, you don't have to worry about the fact that the parts that control sacral muscles versus parts that control arm muscles is mapped, but it is. So there's this map, and I don't want you to memorize it. I'm not asking you to memorize this map, I'm not asking you to memorize this map, and I'm not asking you to memorize this map. I am asking you to know that the anterolateral part carries pain and contralateral pain and temperature information because of the crossing over. I am asking you to know that the dorsal column is carrying ipsilateral touch and pressure information. I am asking you I am asking you to know that there is such a thing as the spinocerebellar tract that also carries proprioception along with the ipsilateral dorsal column. So there's three areas that carry proprioception: ipsilateral dorsal column, ipsilateral, ipsilateral and contralateral spinocerebellar tract. Don't have to know about the crossing over of the spinocerebellar tracts. But you do have to know that they exist and that they're out here, they're lateral. And now I'm asking you to know that this area that we've been leaving sort of blank in our maps, because we were only talking about sensory, is filled with motor information. And it is mostly crossed over. So most of the lateral corticospinal tract, 90% of those axons came from the contralateral motor cortex. 10% or so, well, 8% came from the ipsilateral. And I don't know where the other 2% went. And I'm not asking you to know these, and I'm not asking you to know these, I am asking you to know this. And not for this quiz, but for the exam. Let's take a break. Here's a cartoon of the same thing, right? Contralateral, crossing over in the medulla, and running in the lateral corticospinal tract to or through the area of the spinal cord where it's appropriate, right? So some of this is going to the cervical and controlling arm movement. And some of this is going through the cervical and going all the way to the lumbar, controlling leg movement. Where the fibers end depend on whether they're innervating arm or leg or trunk muscles. Okay, so let's take a break, and then we'll come to some of the higher, we'll go back into the brain and talk about some things other than the cortex. Brain, again. This is from your textbook. So this is vague, sensory motor cortex, but you know that this is the sensory cortex in the post-central gyrus, and this is the motor cortex in the pre-central gyrus, okay? So you know more than the book does. Here's a cartoon of a what's the word I'm blanking on? Coronal section, thank you. Here's a coronal section with some areas of the brain cartoon. And so here's the lateral ventricle, and here this is supposed to cartoon the caudate nucleus, and the putamen, and the globus pallidus. These three areas, caudate, putamen, globus pallidus, form an area called the basal nuclei. Anyway, so we're going to do a little bit of a review of neuroanatomy. Yes?
So is the internal capsule
This is the internal capsule right here, going from the motor cortex. This is not the internal capsule. That's got another name, which I don't remember. This is the internal capsule. This space right here, and right here is the internal capsule. The parts of the brain that we're talking about in the motor system are the basal nuclei and the substantia nigra. So annotate your slide, because I forgot to put it here. The substantia nigra is an important part of the brain that controls movement. Write down substantia nigra here to help you remember that it is substantia nigra and the three elements of the basal nuclei. So some of the motor control in your brain is cortical, and some of it is subcortical. And we're going to be focusing now on the subcortical regions. Actually, we're going to be playing the game. Got a few minutes? Let's play this game, just as a way of reminder. Who wants the laser? You point to anything, and you give a name. Who wants to start?
Charles.
Pedro, you can start. Okay. You started before. Okay, so I'm springing this on you unannounced. I know that some of it is easier and some of it is harder. Let's play this game for a few minutes. Okay? The ones that are in the boxes, which are on your handout, are the ones that are most important. But you can label, if you say, well, I know this one, but it's not boxed. Go ahead, I'll take it, for now. Kendra? Well, you have to say it, so if it's wrong, it's wrong. This box is pointing, so this box is pointing to these two things. What are they? Close, but not quite. Somebody help her out. What is that pointing to? The globus pallidus. Okay?
I don't know what things something.
Okay, good.
Just kidding, go that way.
This one,
Go all the way up
That's the one I was gonna say.
I'm just gonna, corpus
I'm sorry,
callosum.
it's okay.
Okay? It's not conformational change, it's corpus callosum. Good. This line is pointing to the internal capsule. The motor neurons coming from the motor cortex and going to the medulla, which we can't see in this image, and crossing over at the medulla. That is the, say again? Caudate. That's the caudate. It's the what?
Third ventricle.
Show me. Nope, not that one. It's up, the third ventricle's on there, but it's bigger than that. Want to choose something else, or do you want me to show you where the third ventricle is?
Is it that one?
I'm looking over here. Okay. Yes, that is the third ventricle. So I'm not going to draw on the screen over there. This is the third ventricle. Who's got the laser? Marina, have you got the laser? Oh, sorry, Shay. Okay,
But
maybe you can't see it. Maybe it's not labeled. The fornix. So if this is the corpus callosum, the fornix will be under the corpus callosum. So maybe this is the fornix. What's on either side? What is on it? What is the butterfly? Very good. So I think it's this line. It's the lateral ventricle.
This box is singular giant. The singular giant.
Yes. This is the singular gyrus.
Okay. Is it putamen?
It's the, I can't hear you.
Putamen?
Putamen. Yes, the putamen's on there. Which one is the putamen?
Oh, this one.
This one. is the putamen.
Yeah.
Good. Putamen globus pallidus. That's diagnostic.
Is this the hippocampus?
Good. This is the hippocampus. Medial temporal lobe. Hippocampus.
Could this be the thalamus already?
Yes. So this is the thalamus. And I'll give you a hint. The thalamus is labeled more than once on this image because there's different sections of the thalamus. So thalamus is a correct answer again.
Is the hypothalamus right around here?
No, the hypothalamus would be
Oh,
down
under.
here, below
Okay, that's what I thought
hypothalamus. I don't even know if that is, and I think it's not, but because I think the hypothalamus is not in this image.
Alright.
But that's where it would be if it were on this image. Remember, hypo means lower than, so you wouldn't go above the thalamus, you'd go below the thalamus to find the hypothalamus. Let's see.
The midbrain?
Okay, I would have to buy that. I could buy that as the midbrain. So, yeah, let's, this might be the mammillary bodies. It might, I'd have to look in the book to see what was actually labeled, but I would buy that as midbrain. Let's do 2 more boxes. Let's make sure we get the boxes. So, what is this box or this box? This, no, this, or this on this side? Yeah, it's part of the thalamus. So this one is also thalamus. And I'll give you the last one, the other one. This one is also thalamus. Remember, the thalamus has lots of sections, right? We've only talked about one section of the thalamus called, well, no, we've talked about 2. The lateral geniculate nucleus and the medial geniculate nucleus of the thalamus. So in this thing, they were labeling different nuclei of the thalamus. But that's more than I'm asking you to know, but I'm asking you to know those. And let's see, does anyone want to venture a guess what this is? Well, this is the lateral sulcus, or the lateral fissure. What is this thing that is pointing to? Because it's not pointing to the sulcus, it's pointing to this gyrus. It's the auditory cortex, or really it's the superior temporal gyrus, right? And that is where the auditory cortex is. Okay, so we could play this game for the rest of the period, but we're not going to. The answers are here. Did we get everything right? Yes. And the ones that are in yellow are the ones that are part of the motor system. Specifically, the basal nuclei, which are caudate, putamen, and globus pallidus. The thalamus is I guess I didn't say this before, but the thalamus is a relay station for motor information, and similar to the way it's a relay for sensory information. And what's not on here, but you might want to just write the word down again on this, is the substantia nigra. That's also, it's not on this image, but that's part of the motor system. Questions about this anatomy? I will not ask you these kinds of anatomy questions on the quiz, but I could ask you this kind of anatomy question on the exam because it's motor system anatomy. So here's a cartoon of the parts that we're talking about. Globus pallidus, putamen, caudate, parts of the thalamus, substantia nigra, which I don't think was on, this is cartoon, and what is called the subthalamic nuclei, which are, different, another part that is below the thalamus. But the most important remember we've got cortical control, cortical regions in the motor cortex, in the pre-central gyrus of the frontal lobe, and we have these subcortical regions: caudate, putamen, globus pallidus, substantia nigra, thalamus. So there's a program that we run in our brain. And this is interesting because I'm going to get to Parkinson's disease. So, just like there is a motor program in the spinal cord where we have inhibitory interneurons that make sure that when we contract a flexor, we relax an extensor. That's due to inhibitory interneurons. We have inhibitory interneurons that are important in this motor control. Basically, the subcortical regions, the thalamus, the putamen, the globus pallidus, the substantia nigra, the subthalamic nuclei, all the things that we've been focusing on for the past few minutes. This is what's important for initiating movement. So when you think of moving your arm, there's actually subcortical regions that are working before you actually execute the movement of your arm. They've timed this out. There's activity in the subcortical regions 100 milliseconds before there's activity in the cortical region for a voluntary movement. What you think of being voluntary isn't voluntary until after your subcortical regions have done some work on it. So this is a complicated map, but I want to illustrate an important point here. And that is inhibitory, inhibition of inhibition is a thing here. So let's look at this. We've got the substantia nigra. Blue, in this case, blue is an excitatory neuron, and red is an inhibitory neuron. The substantia nigra is critical for initiating movement. And someone with Parkinson's disease has a damaged substantia nigra. So that's why these lines are fine, because they've been damaged. There's been apoptosis in the substantia nigra. The substantia nigra is smaller and less active. Well, the cells have died. The cells in the substantia nigra have died in Parkinson's. And so that's why we have robust signaling from the substantia nigra to the putamen in normal, and we have weak signaling from the substantia nigra to the putamen in a Parkinson's patient. Okay, so that's what we're illustrating here. So let's look at this blue one. Blue activates receptors, dopamine. This is a dopaminergic pathway. Remember, substantia nigra is the home of dopamine-containing neuron cell bodies. Dopaminergic activation, it's excitatory, dopaminergic activation of a neuron that is probably GABAergic because it's inhibitory. So this neuron inhibits this neuron, and this neuron inhibits this neuron. And this neuron in the thalamus excites the motor cortex and says, okay, I'm ready, I have initiated, I'm ready to go. Initiate voluntary movement. Okay? So inhibition of inhibition allows excitation. A double negative. You start with a coin on heads, you inhibit it, You inhibit it again, you've got heads again. Multiply something by -1 twice, and you're positive again. So inhibition of inhibition allows the thalamus to excite the cerebral cortex, and we have voluntary movement. We can do the other pathway, inhibition of inhibition of inhibition of inhibition. 4 -1 to the 4th power is a positive number. So let's not go through it too much, but remember, if inhibition of inhibition is excitation, inhibition of inhibition of inhibition, excitation, inhibition is another excitation, and that's, well, again, allows the thalamus to excite the cerebral cortex. What do you need to know? That the substantia nigra controls the putamen, and that there is inhibitory and excitatory pathways in this circuit. And knowing that inhibition of inhibition, we'll just stop there, inhibition of inhibition allows these neurons to be excited. This neuron inhibits this neuron, but this neuron inhibits this neuron. So exciting this neuron inhibits the inhibition, allowing excitation of this pathway.
So, these are working simultaneously?
Yes, they are.
So for the red one, they'll also
This one?
Yes.
So it's also going to the putamen.
Yeah, so you have inhibition of inhibition, so that's excitation of the inhibitory.
So inhibition of inhibition, so it's exciting this.
And so if this is
Well, wait.
excited,
No, this is, so it gets complicated. So this is inhibited. So this is, when we initiate movement, this neuron is excited. It's excitation of an inhibitory neuron. So this is excited, I think, I haven't worked this through recently. Let's just see if that, let's not do it. I don't want to do it in front of everybody because I could make a mistake and make things confused. It's, when we get with 2, it's easy. When we get with 4, it gets complicated. Yeah.
So on the right side, there's a bigger.
Okay, so now let's get to Parkinson's. Let's do just the blue pathway. We have less excitation of this inhibitory neuron. So this inhibitory neuron is not inhibiting this neuron. So this neuron is maximally inhibiting this neuron. And so we have less excitation of the cerebral cortex. And we have Parkinsonian rigidity. Some of you may know that the symptom of Parkinson's disease is immobility, the inability to initiate movement. Muscles are fine, in most cases, most of the brain is fine, but the ability to initiate movement is impaired because we don't have the substantia nigra getting the ball rolling in the basal nuclei, telling the cerebral cortex to go, to move, to create the voluntary movement. The person wants to move, but they cannot initiate the movement. And they develop what's called Parkinsonian rigidity. Yeah,
why is that one still inhibitory though if it's?
This one?
No, the one in Parkinson's,
yeah.
it's still inhibition and
So it is inhibition of inhibition, but we have inadequate activation of that inhibition of inhibition. Does that help? The other part, so the last 5 minutes, we want to talk about the cerebellum for just a minute. So the cerebellum is an important part of where we control fine motor movement. So those of you who play guitar might know that you have memory in your brain, in your cerebellum, and in your spinal cord that helps you control movement of the fingers for playing the guitar, or for throwing a great baseball, or for, so both fine and gross motor movement is coordinated in the cerebellum. So we could spend a whole course on cerebellar inputs and outputs. We could spend a whole course making sense of this pathway. And I'm not going to do it, but I'm going to spend 3 minutes doing it. So you'll remember, because it helps me remind you of certain things that you do know. Remember that the spinocerebellar tract is providing information to the ipsilateral cerebellum. But you also know that it's the contralateral cortex that is most important for certain muscles, so for controlling certain muscles. So the muscles of my legs are providing proprioception of my right leg. No, my right leg is providing, well, let me do left leg. The proprioception of my left leg is providing information to my left cerebellum, but it's my right cortex that controls my left leg. How is that possible? Well, you can see there's lots of crossing over between the cerebellum and the cortex. So here is a afferent pathway, input to the afferent to the cerebellum. Ipsilateral, lots of processing. Okay, so now we're going to just, the red is on both sides. But now we're still ipsilateral. The output, ipsilateral output, crosses over to the thalamus, contralateral, and to the contralateral cerebral cortex. So spinocerebellar input, processing ipsilateral cerebellum, and then outputs to the contralateral cortex. The cortex provides information to the cerebellum. We've got sort of an error minimization program. Our cortex says, I need you to run fast enough to catch that ball that is in this arc. The cerebellum is doing this complicated calculation based on visual information, based on proprioception, how fast am I running, how fast is that ball moving, how fast do I need to run in order to get my mitt on that ball out in center field. The cortex is saying, do it, but the cerebellum is doing the calculation. And that's the importance of the cerebellum. I mean, that's one of the important bits of the cerebellum. Ipsilateral input from the body, contralateral input from the cerebral cortex. Comparison, okay, how fast am I moving? How fast do I need to move? Move faster or move slower. Do I need to run more or do I need to put the mitt up, right? Ipsilateral body information, contralateral cortical information, processing, and then output to the contralateral cortex to make the adjustments necessary. Okay, that's it for today. I'll see some of you in lab tonight.